This is a muscle fiber, also known as a muscle cell. Each one of our muscle cells is made up of several strands, known as myofibrils, and then each myofibril is made up of several units of sarcomeres. Now, each sarcomere has two very important protein filaments called myosin and actin. So then, how does this relate to muscular contraction? The answer is the sliding filament theory. You see the myosin filaments? They have these golf club-like extensions coming off from the body. Now, these extensions love to latch onto the binding sites of the actin protein. When this happens, the cross bridges use a maneuver known as a power stroke that is powered by the energy adenosine diphosphate plus an extra phosphate, ADP plus P. Once they latch on, they slide the actin filament towards the center of the sarcomere. So here, in this model, the myosin cross bridges are going to attach onto the actin filaments, and they're going to slide it towards the center. This continuous sliding then brings the actin closer and closer together, reducing the diameter of the entire sarcomere. As all the sarcomeres begin to compact, then the myofibrils that they're housed in begin to compact as well. Then the whole entire muscle fiber begins to contract, when then the entire muscle begins to contract as well. However, the actin has these extra two additional proteins that surround it that block the binding sites from the cross bridges. These proteins are called tropomyosin and troponin. When the brain sends the signal for the muscles to contract, it tells the sarcoplasmic reticulum, a membrane that surrounds each myofibril, to release calcium. This calcium binds onto the troponin, which then changes its shape. When the troponin changes shape, it moves the tropomyosin out of the way and leaves the binding sites exposed for the cross bridges to latch onto. Once those binding sites are exposed, the sliding filament theory can continue. And so it latches on, and then it pushes the actin filament towards the center of the sarcomere. Then, in order to relax the muscle, all the brain has to do is send a signal. It soaks up the calcium back to the sarcoplasmic reticulum, and then the troponin changes back into its original form. When the troponin changes form, the tropomyosin covers the binding sites again, and the cross bridge is no longer able to attach onto the actin. Without muscular contraction, our legs wouldn't work and our hearts wouldn't pump. But thanks to the sliding filament theory, our muscles allow us to get up and engage with the world physically. So remember to appreciate your muscles, because without them, we would cease to exist.